Afternoon, everyone. I just wanted to welcome you to our first session of our PMSF 2022 virtual conference. My name is Denise Croden, and I am a member of the board and also a member of this incredible conference committee who has brought the first session to you. Hopefully you guys are ready for an exciting session. But first, I want to kick off a commercial from one of our sponsors.
Our mission at Jaguar is to accelerate breakthroughs in gene therapy for patients with severe genetic diseases.
With gene therapy, we can give the opportunity at a better life. That's what makes Jaguar special.
Without further ado, I am excited to introduce Dr. Kate Still, who is our scientific director, and she's going to be moderating our genetics session with some fabulous contributors, which I think many of you already know. But Kate, take it away.
All right. Hi, everybody. It is so great to be here with you all. Welcome, everyone, to this genetics session, which we thought was appropriate for the start of our conference. Because it represents the start of diagnosis, and also there's a lot of interesting science going on in this area. A couple of housekeeping notes. We will be saving questions for the end of the session, so feel free to post your questions in the chat as you have them, but we will field them at the end. So let me introduce Thomas. It is my absolute pleasure. He is a professor and head of the Human Genetics and Cognitive Function at the Université de Paris, Institut Pasteur in France. Professor Bourgeron has done groundbreaking work in identifying a set of genes which are highly associated with autism. Among these is SHANK3, which we know plays a very important role in PMS. He has done foundational work characterizing animal models of autism and PMS and is involved in large studies across Europe, focused in part on people with PMS. He's an active member of the PMSF Scientific Advisory Committee, and he's a passionate advocate for people with autism and PMS. In the interest of time, I can't do him complete justice, but welcome, Professor Bourgeron, and I will pass the ball to you for your talk.
Thank you so much, and thank you. I mean, it's a really an intense pleasure to be here with you. So I will share my screen and start the presentation. Okay, great. So thank you again. That it's wonderful to be here and from Paris, but with you. And so my talk will be on genes, mechanisms, and possibly treatments for Phelan-McDermid syndrome. So just to say that the door of my lab is at the hospital, and we are working hand in hand with the clinicians. So I'm really lucky to work with amazing clinicians in Paris by the team of Richard Delorme. And the clinicians are geneticists, psychiatrists, psychologists, working and trying to understand what's going on. And they also share the data with us to do more genetics, more brain imaging, and we also do mouse model in the lab. So my lab is at Pasteur in Paris, but and I also work on a lot of groups in Europe through different EU-funded projects. So this is our idea of what could be the research for Phelan-McDermid syndrome. First, we need to identify, because if we don't identify the right cause, we might be on the wrong tracks. And then to understand what these causes are doing, and then if we understand well, can we intervene? And so the strategy is this one. You have people with autism, because that is the broad project of my lab. So we are doing a lot of work on genetics, trying to understand what are the genes involved in these conditions. And then when you have a set of genes that are identified, then you can do cell models, mouse models, animal models to better characterize the role of the gene that is functioning differently in people with autism. And then you can start to understand what are the mechanisms and what are the molecules that could reduce the the adverse symptoms caused by these mutations, and then to do targeted clinical trial. And I have to say, for Phelan-McDermid syndrome, we are doing all the steps are there. So we identify the one of the major genes, SHANK3, then we can do cell models, we can do animal models, and do the screening of molecules and starting the targeted clinical trials. So let's start with the genetics and why I came to Phelan-McDermid syndrome. This is because in 2006, we found this gene called SHANK3 that you probably know very well on the tip of the Q arm of the chromosome 22. And this gene is involved in autism, but is also a major gene, as we will see, for Phelan-McDermid syndrome. And this protein coded by the gene is in a network that is also associated with other genes associated with autism. And this protein network is at the synapse. So the synapse are the contact between nerve cells, and where is SHANK3? It's there. So it's really like a scaffold of the synapse, the glutamatergic synapse in the brain. Okay, so one questions that I usually have from the families is why SHANK3 is an important gene for Phelan-McDermid syndrome. But also, I have to stress also that SHANK3 is a major gene, but we also need to see the bigger picture. I mean, every time we need to see the bigger picture. So I will talk about SHANK3 and other genes. So as you know, Katy found this Phelan-McDermid syndrome. She will talk after. And why SHANK3 is interesting, because in most of the deletions, you know that there are large deletions of 22q13 region, sometimes very large, sometimes very small, and they almost all include SHANK3. So that's why SHANK3 is a major gene. I mean, this is one of the reasons why. But also, Katy found some, and others found some interstitial deletions that apparently do not affect SHANK3. So we have now a very simple classification, say PMS SHANK3 related when it's a deletion that remove part of the gene of SHANK3 or a single mutation of SHANK3, and PMS SHANK3 unrelated when the deletions are not affecting, apparently not affecting the SHANK3 regulation or the SHANK3 gene. So why SHANK3 is an important gene for Phelan-McDermid syndrome? Because so in 2006, we found mutation of this single gene in patients with autism and with intellectual disability. And more recently, for example, with the group of Steve Scherer, in Toronto, we found a mutation in the SHANK3 gene. So this is the SHANK3 protein here. And you have this mutation that is found in several patients. This is a de novo mutation, so the parents don't have the mutation, and you have the mutation. And we could find this mutation in something like more than 12 patients. Most of them, they have autism. Most of them, 80%, they have intellectual disability or global developmental delay. Speak language was also, in majority of the patients, there was absence of or some language delay, and other neurological and psychiatric comorbidity. So as you can see, a single mutation of SHANK3 recapitulates most of the symptoms that you have that you can observe in patients with Phelan-McDermid syndrome. But so SHANK3 is super important for sure, and we need more research on SHANK3, but we also need to see the bigger picture. And why? Because as Katy Phelan showed a long time ago, there are a couple of deletions, and SHANK3 is not always the only gene in PMS. And as you know, the patients with Phelan-McDermid syndromes, they can share some symptoms like intellectual disability or some of the speech problems, but for some other symptoms, they are very different. Some are very severely affected in their motor function, some have epilepsy, some have other aspects. So we need to understand why these individuals have a different clinical trajectory. And this is my slide in French for Ronnie and Kate. And so this is super important. So we need more information on these comorbidities. And so I urge you to go to the international registry from the Phelan-McDermid Syndrome Foundation, because this is super helpful for us to understand. So if you want to go to the French website, you can, but you can also go to the website from your country. And then if we have some information about the different clinical symptoms, you can start to understand what are the genes which are related to specific symptoms. So this is a study that we conducted with Anne-Claude and Thomas in my lab. So we have patients with very large deletions and patients with very small deletions. And then this is a very busy slide, but let me explain. We look like a sliding window and we say, for this part of the genome, what are the symptoms of the child? And so take an example here. You have, if you take the patients with Phelan-McDermid syndrome, you can say that 50% of them will have a language absence of speech. But this is not always the case. If you look at the deletion, if you take very large deletions that goes like more than 8 megabases, then 80% of the children will have will observe an absence of speech. But if you take the small deletion, you have only 20% who have a problem, absence of speech. They can speak a little. So that's super important. So probably you can do this for many aspects. So absence of speech, ophthalmic features, for example, or other aspects. And then you can start to map genes that, in addition to SHANK3, can also reduce, for example, the development of the speech. But as you can see, so that's what we can do at the 22q13 locus. But as you can see, the epileptic seizures, you can see that the patients with very small deletion, they can have epilepsy, and the patients with large deletions also. So it's not as clear as, for example, for ophthalmic features, where most of the patients with ophthalmic features are in the large deletion. So you have to go further. So this is a chromosome 22, and you can see that it's a patient. So each year it's a patient. And you can see here, it's a patient with a large deletion, it's a patient with a small deletion, and so on and so on. And so this is the chromosome 22. But then you can look at all the genome, the bigger picture. And what we have seen is that other variations were present. And for example, this child here, he has a 22q13, he has a loss of SHANK3, but he also has a 16p deletion, which is another locus for autism. So this is a very complex slide. I don't want to go into detail, but just to give you the way we are thinking and the way we are doing our research. SHANK3 is super important, but we have, between you and me, we have 3 million of genetic variants. So the idea is now to understand what are the variants that could explain why a child has autism, why he doesn't have autism, why this person has epilepsy or regression. And so we are doing now this using the whole genome sequence. And so the idea is really to understand what is the bigger picture? What are the genes that contribute to the comorbidities like epilepsy or regression and so on? So this is the first step, to identify the cause and then to understand what's going on. And for this, you can look at the brain. So this is what we are doing. And for example, we could see that some patients, and you have normally this corpus callosum that makes the link between the left and the right brain, which is missing or very small in patients with Phelan-McDermid syndrome. But as you know, it's very difficult to go inside the brain in humans. So we need animal models. And that's why we met with Tobias Bockers, met this shank-free knockout mouse. There are a lot of knockout mice for shank-free. And what we wanted to see is the behavior of these mice. So we developed recently new methods that are the really cutting-edge methods to really understand the behavior of the mice. So everything that the mouse is doing, it's sent to a database. So now we have a really comprehensive idea of the behavior of the mice, of the social behavior of the mice during long term. So you have all this behavior, and this is, for example, a very simple behavior, but just to show you an example. This is a wild type mouse, and we will put an object like this here. And you see that the wild type mice, we look at the object and we'll have this stretched posture to look at the object. In the SHANK2, this is another gene that we are exploring in autism. The mice is hyperactive. You can see it's super active. And when we put the little house, it goes inside the house like this. And look at shank-free. Shank-free, they are hypoactive. And when we put the box, it looks like the mouse is afraid of the box and doesn't want to go inside the box. So now we have some tools to really look at the behavior of the mice and trying to find new molecules that could reduce the deficits observed in the mouse. And so one of this is a shank-free mouse here. And one of the obvious phenotype is this grooming behavior. So we could quantify the self-grooming, and this is highly significant in males and in female mice. They're really grooming a lot. And to make a very long story short, that is not yet published in a regular journal, but we made a lot of transcriptomics and so on, and we found that one part of the brain, the striatum, is very different. I mean, very different. It's different in the shank-free knockout with a striosome. So this is a part of the striatum. So for the aficionados of the striatum, I can go into much more details. And this is the work of these wonderful people here. So now to intervene, my last part, and that's probably the one you're waiting for. So let me tell you about this project. So then we have this mouse model, but we also looked at neurons derived from the cells of the patients. So the team of Richard Delorme, the clinicians, they went to the patients, they sampled blood or skin samples, and then we derived these cells into neurons. So we have the neurons of the patients, and we could see that the neurons of the patients actually have less synapse. This contact between nerve cells were reduced in the patients. And with Alexandra Benchua, she took the cells and she screened for 205 molecules, FDA-approved molecules, so medication that is already in the market, to see if there is a molecule that could boost the shank-free expression. So you know that most of the kids are lost one copy of shank-free, but they still have one copy of shank-free. So the idea is to boost the copy that is still there. And one of the molecule that we found, compound, is the lithium. And so why lithium is important for PMS? So when we published this result, a really amazing neurobiologist from US Ginato and Giano, looked if the shank-free had a problem, that the synapse, she could find that there is a problem. I will not go too much in details, but she found that if she add lithium, she could reverse the phenotype, and then she also could reverse the time spent grooming. So it's really a good. We are not impatient again, so we have always very good news for the mice, and unfortunately not very good news for the patients. So, but this is a first step. And Tobias is really, I mean, we are working for more than 10 years together. And when I said to him, can you try lithium in on the neurons? And he came to me and he said, that's fantastic, with a German accent. And he said that because it really boosts the shank-free in the we So that's, again, this is in vitro in the lab. So can we have some clues that it could be efficient in patients? It's really the beginning. So there are some reports, like in 2 patients here, they found that it could rescue the regression. This is our patients where we have a single patient, so really n equal 1. So it's really not a proof, it's really a proof of concept. And there is also other report in the literature. But what we need, which is super important, is a randomized controlled trials. As you know, this is really important to be sure that the molecule is doing this. So if you speak French, like Ronnie, and maybe Kate also, you can say that this is the effect of lithium, and this is a pilot, this is the RCT, a randomized controlled trial that we are doing now in Paris. So we will test 22 children under 18 years old. They will be treated with lithium or placebo during 12 weeks. And the primary outcome that we will look is, are they better on the social responsiveness scales? And we have secondary outcomes also. And the first patient was included in March. So this is really the beginning. So hopefully I will give you more info in the near future. So last, this is the also a big special thanks to Connie, who is leading, and you will have very soon a European guideline for Phelan-McDermid syndrome and a new database for clinical trial for patients with Phelan-McDermid syndrome in Europe. So this is really almost my last slide. So this is the genetics, how I see the genetics. So you have strong alcohol or light alcohol, and you have this kind of, and the same for the genes. You have some mutation that will have a large effect and some mutation that will have a small effect. And let's take this picture. If you remove this person from the orchestra, you will hear a difference of the song. If you remove this guy or this guy, unless you have a very good ear, you will not make a difference. But if I remove all these guys, probably you will see the difference. So this is Madame Shank-free, and these are all the other genes. So we have to understand the complete orchestra and the different genes that are important in the syndrome. So that's all the people that I want to thank, because I'm talking, but the people are working in the lab, and I'm really so lucky to have all these people around to collaborate or to work with. The funding, and I would like to thank a special thanks to the Phelan-McDermid Syndrome Foundation from France and the TEANI Association, because they provide funding and they provide IDs, and we are working together, and this is amazing. Voilà, merci beaucoup. But if there are questions, I will be super happy to answer.
Thank you so much, Thomas. We have already received a few questions, and we will do our best to send them to you later or try to answer them at the end of the session. Thank you for an excellent talk. And I really enjoyed especially the last slide with that picture of, understanding how different genes can contribute. And I think a lot of families can recognize that a lot of the papers we have coming out recently are this concept of genotype-phenotype correlation, or changes in genes and how that impacts symptoms. And so that was a great summary for everyone, and we really appreciate it. So rolling right along, up next is Dr. Curtis Rogers. He is the senior clinical geneticist at Greenwood Genetics Center. He has evaluated many people with neurodevelopmental disorders and intellectual disability, and he's a leading genetics expert in PMS. He was actively involved in originally identifying and describing the syndrome alongside Drs. Phelan and McDermid, and he has been a huge support ever since. He has served as a founder and chairman of the PMSF, and he's an active member of the Medical Advisory Committee and advises many PMS families on genetic testing and clinical concerns. Welcome, Dr. Rogers.
Yes.
Okay, so let's get started. I'm going to talk a little bit about genetic testing and diagnosis, because in order to get to Thomas's research protocols, we obviously have to make the diagnosis, and we have to have a high suspicion of that diagnosis in clinical patients that we are seeing in our offices. You can see we're going to talk about the incidence briefly, the features, how we do genetic testing, and what different genetic alterations there are in PMS. I will tell you this won't break down exactly like that as we go through the slides. Thomas was talking about autism and separating out people who have SHANK3 variants from people with autism who have other variants, and they are looking at synaptic genes. And let me just say, there are probably 1,500 different proteins involved in the function of the synapse, every synapse in your brain. Therefore, there are 1,500 genes encoding those proteins. So obviously, many other genes could be important in the presentation of a child with autism, and specifically a child or adult with Phelan-McDermid syndrome. But if we take the autism data and what we found in that, and we realize that most of the folks who have Phelan-McDermid syndrome are going to have deletions of 22q13.3, we come up with an incidence of 1 in 5,000 to 1 in 10,000. Now, obviously, we don't have that many people involved in our database or registry yet, and that's because many people have not had testing yet. And until we test everybody and have a much more large-scale study, I don't think we'll know the exact incidence. What are we looking for clinically when we see a patient in our office? We're looking for hypotonia. So baby that is somewhat limp, normal growth in most. Many will have delayed or absent speech, especially early in life, and that speech may be very delayed. They may develop speech and then lose speech. Global developmental delay, so they're going to have problems not just in speech but in motor skills also, and some minor dysmorphic features. You can see on this little fella here, he's having to be held up because he has low muscle tone and decreased strength. I think you can appreciate he's got kind of puffy hands and feet, a little bit of a round puffy face, mild droopiness of the eyelids, what we call ptosis, a little bit of a broad nasal tip. And if you look at the toenails, they are small and dysplastic and peel. Many of our parents have commented on their kids' toenails, who should be tested, and we'll get into how we test them. But if you have another genetic diagnosis and they haven't been able to confirm that, so, oh, we think this child has Angelman syndrome, but the testing is negative for Angelman syndrome, or they have autism, which is a highly diverse etiologic diagnosis, things like that. If we can't find the cause, we certainly need to consider making sure that we've ruled out Phelan-McDermid syndrome. abnormal chewing behaviors, and many of the parents will tell you that they use industrial strength chew toys for their kids because if not, they will chew on anything. Uh, Autistic behaviors may include poor eye contact or tactile sensitivity, repetitive behaviors, and any child with hypotonia with delay, I think certainly needs to be proven not to have Phelan-McDermid syndrome. A little bit about genetics. Your genome, which is made up of 3 billion, that's with a B, base pairs are letters of DNA, and you can see the alphabet of DNA is A, T, G, and C, but 3 billion of those letters that are organized onto chromosomes, which are made up of genes, which are made up of these individual letters. So you can think of the story of your life genetically as the book is the genome, the chapters are the chromosomes, and the sentences are the genes. If we look specifically at this ideogram or this cartoon of a chromosome 22, you'll see how we name chromosomes, a short arm above that little indention called the centromere. It's called the P arm, and the bottom part is called the Q arm for the long arm. And you can see in 22q13.3 deletion or Phelan-McDermid syndrome, what you see is this deleted segment enclosed in that box that is typically there. That deleted segment can be extremely variable from a small deletion, 130,000 or even less than that KB. KB means 1,000 base pairs to over 9 megabases or 9 million base pairs. And you can imagine, as Thomas was discussing, how many genes could be contained in that area. But as he said, there are also genes located, we're talking about one gene, SHANK3, that leaves us still with 20,000 genes that may be contributing to the phenotype that we see, and that are located not just on 22, but are also located on every one of the chromosomes we have. The phenotype thus far is not highly correlated. You saw, Thomas, with some charts showing, well, the seizures not so much, maybe the GI symptoms, yes. Maybe autism, yes. Maybe speech problems, yes. So we're still working on that to try to determine that. He did mention the critical gene appears to be SHANK3, but that's not the only gene. If you look at how Phelan-McDermid syndrome occurs, and I'm just focusing right now on the deletions at the end of the chromosome, we in genetics call those terminal. I do not like that name. It does not mean a child is going to die because of this deletion. And I wish someone had not named it that. But at the end of the chromosome is where we're talking about right now. You can see that roughly 80% of individuals with Phelan-McDermid syndrome have simple deletions. Some will have an unbalanced rearrangement. We'll talk about that in a second. And ring chromosomes that I think Katie is going to talk about a little bit. Mosaicism thus far appears to be unusual in Phelan-McDermid syndrome, at least as far as we know. This is a 1 cell's worth of chromosomes. We call it a spread because you get this mishmash. Then we take this, and very smart people like Katy Phelan will take this and organize this into a recognizable pattern we call a karyotype. And in this karyotype, what you see is that the bottom of the 22 is missing where that green arrow is. You can see that that 22 is shorter than its other 22, indicating that there's a deletion present there. You'll also notice that that little dark band at the bottom of the 22 on the left is not present on the 22 on the right, again, indicating a deletion. So this is what we would see, and you can see that that band is missing, the dark band and the light band at the very tip of that normal 22 chromosome are deleted or missing. As Thomas showed you, there are many different sizes in deletions that go from the smallest size on your left of this slide all the way up to much larger deletions, 8, 9, 10 megabases to the right of that slide. We can use studies like FISH, fluorescence in situ hybridization. You'll notice on the normal chromosome 22, there is a red probe that is a marker probe present on the 22. We use that to find the 22s, and then we use a specific probe for 22q13 that is shown in green, and that lights up on the normal chromosome because that piece of the chromosome 22q13 is present, whereas in the deleted chromosome, you do not see a green fluorescent probe indicating that that piece is missing from the end of the chromosome. I mentioned simple deletions occur in about 80%. Most of those deletions occur on the father's chromosome 22. They are de novo in almost all the cases. However, just like in many other chromosomal deletions, it appears that the father's chromosome 22 is more susceptible to a break than is the maternal 22. I think that brings up an interesting question as to whether there may be a difference in individuals who have Phelan-McDermid syndrome if the deletion occurs on the paternal 22 or whether it occurs on the maternal 22. Translocations can occur in about 10% of individuals with Phelan-McDermid syndrome. 80% of those are inherited from a parent who has a balanced translocation, and about 20% of those are de novo or new variants on the chromosomes. Inherited translocations pose a much higher risk for recurrence in subsequent children for a couple if one of them is carrying a balanced translocation. So in an unbalanced chromosome translocation, we actually lose genetic material, whereas the balanced part of when we say a balanced translocation means that we have not lost or gained any genetic material. When it's unbalanced, we've lost and probably gained some material from another chromosome. If those two pieces, when you're looking under the microscope, are roughly the same size and have that same banding pattern, that light and dark band, it may be very difficult to see. And that's when FISH would become very valuable in helping us see that, and also a microarray. So what you see here is a balanced translocation. Part of the green or aqua chromosome on my screen has, from the chromosome 4, has moved over to chromosome 22. 22 has had a break, and it's exchanged the bottom part of 22 with the number 4 chromosome there. And this could involve any chromosomes. What you see here is an unbalanced translocation because we have gained some, that slide is mislabeled. I just noticed the smaller chromosomes are 22s, and what you can see is their material from the aqua chromosome, which is the number 4, excuse me, that is at the bottom of the chromosome 22, and it replaced material that was lost at the bottom of the purple chromosome or the number 22 chromosome. I'll have to fix that. You can also have a ring 22, which is another 10 to 12% of individuals with Phelan-McDermid syndrome. And what you can see in there is that there's been a break at each end of the chromosome. The chromosomes don't like those sticky ends where there's been a break, and they tend to form into a little ring like that because you've lost material at both tips of the chromosome. that ring is unstable during cell division and can lead to additional problems besides Phelan-McDermid syndrome. This is a picture of a microarray result. And what we've done with this microarray is this is all done on the computer. And what you can see over to the far right of the slide is the fact that the purple area is down lower than the general reporting for the chromosome 22. So what we have down there is it's dropped down to the -1 dotted line, indicating that that part of the chromosome is deleted. There is not 2 doses of that. There's only 1 dose of that particular area of the chromosome, indicating that we have a normal 22 and a 22 with a deletion at the tip. The other way we can find this, as Thomas indicated, was looking for pathogenic variants or mutations within the SHANK3 gene that would be likely to present with symptoms such as autism, and many times other symptoms that would be consistent with a clinical diagnosis of Phelan-McDermid syndrome. That is going to require typically either targeted testing for a SHANK3, or many times how we're going to find that is with whole exome sequencing or on an autism gene panel test, where we look at all of the genes or certainly look at a large group of genes to see if there are any variants in those genes that could explain that child's autism, intellectual disability, and other symptoms that they may have. So for SHANK3 related Phelan-McDermid syndrome, we're going to need a disruption of 22q13.3 that includes the SHANK3 gene or disrupts the SHANK3 gene, or we're going to need a pathogenic variant within the SHANK3 gene. As Thomas also showed you, we have now come up with a classification system where we have broken Phelan-McDermid syndrome down into SHANK3 related and SHANK3 unrelated, whether it are associated, whether it includes the SHANK3 gene. So I will stop there, and I think I got through in 15 minutes, and we'll see who is up next.
Thank you so much, Dr. Rogers. That was extremely educational.
That's pretty quick talking for a Southern boy, I'll tell you that.
It was fantastic. And I know that we received a lot of questions along those lines, and that your talk generated some questions in the chat and the Q&A. So thank you so much for that. And next, it is my pleasure to introduce Dr. Katy Phelan. If you don't know of Dr. Phelan, you will immediately recognize her name as the namesake of our syndrome. Dr. Phelan has an extensive experience in genetics and originally identified and reported on a case of 22q13 deletion in 1988 and has continued to characterize the syndrome ever since. She was instrumental in growing what was the family support group into the Phelan-McDermid Syndrome Foundation alongside Dr. Rogers, Sue Lomas, and Nick Assendelft. Dr. Phelan is the Director of Genetics at the Florida Cancer Specialists and Research Institute, but she has also served as Director of Cytogenetics at several other institutions, including Greenwood Genetics Center. Dr. Phelan has served as founder and director and is an active member in the Scientific Advisory Committee, where she continues to advocate for families. Thank you for joining us, Dr. Phelan.
It's great to be here. Somebody's going to work my slides for me, right? Yes. Okay, let's go to the first slide. I'm going to talk about first the classification system that Thomas and Curtis have mentioned. Hey, Andy. As you can see on this paper, there are 14 authors here, and the authors are researchers, clinicians, lab scientists, and parents who are also researchers, clinicians, and lab scientists. And the expertise here is expertise in the clinical identification and characterization of deletion 22q13 before it was named Phelan-McDermid syndrome, and the discovery and characterization of SHANK3, and expertise in parenting individuals with Phelan-McDermid syndrome. So we have a lot of expertise here. We have over 30 years of experience, as we say. And I don't think we can question that the authors here are qualified to make this classification system, which we did. And we represent here the Scientific Advisory Committee and a few members of the Medical Advisory Committee of PMSF. So let's go to the next slide. I included a timeline here that led us to the classification system. So the first time point in 1985, there was a recombinant chromosome 22 that had a deletion of 22q13 that was described. And then in 1988, the case that Curtis and I described in a newborn male that we saw at the Greenwood Genetics Center. And about this time, about a half a dozen individuals between 1988 and 1991 were described with deletion of 22q13. And then in 2001, there were a couple of studies that said that SHANK3 was a major gene in the 22q13 deletion syndrome, and it was deleted in most individuals with the syndrome. And as Thomas told you, SHANK3 is a very important gene that's expressed in the brain, and it really explains most of the neurological features that are expressed in Phelan-McDermid syndrome. Then in 2002, the syndrome was named Phelan-McDermid syndrome at the urging of the parents who didn't like their syndrome named by a number, 22q13. They wanted a name associated with it. And then in 2006, 2007, the SHANK3 gene was associated with autism spectrum disorders, which was a great tie-in with us because our children have autism and autism spectrum disorder. So it was a great fit with Phelan-McDermid syndrome. And then in the years following this, there were questions about interstitial deletions. Were they associated, were they really Phelan-McDermid syndrome, or did you have to have a loss of SHANK3 to have Phelan-McDermid syndrome? So these questions were kind of pervasive. And I mentioned this one study in 2014 where there were 9 individuals with interstitial deletions, and the authors were proposing that this was a new syndrome, not Phelan-McDermid syndrome. So we got together and published this manuscript, this classification system that I will describe on the next slide. So the classification system is that there's PMS SHANK3 related and PMS SHANK3 unrelated. And so SHANK3 related is there's a presence of a deletion or disruption of 22q13 that affects the exons or the coding sequences of the SHANK3 gene, or there's a demonstration of a pathogenic variant of SHANK3. So we used to say a mutation of a gene, now we say a pathogenic variant of a gene. So if you're confused by that term, that's what we used to say mutation, but the American College of Medical Genetics wants us to say pathogenic variant. And SHANK3 unrelated is that there's an interstitial deletion of 22q13 that does not affect the promoters or the exons of the SHANK3, although the main features of PMS are present. So if you had an interstitial deletion of 22q13, but you didn't have the features of PMS, then you don't have PMS. So if you had a deletion of 22q13 and you had normal language, polydactyly, and club foot, those aren't features of PMS, so you don't have PMS. You have to have the features of PMS to have PMS, okay? That makes perfect sense, right? Okay, the next slide, please. So the system is straightforward. It's very simple. It makes a lot of sense. It is inclusive. It takes into account the fact that the people with the terminal deletions, which is the end of the chromosome, the term that Curtis doesn't like, but it means the end of the chromosome, and the people with the interstitial deletions have the same features. It includes all of the families, right? It resolves confusion for clinicians that have been confounded by the fact that suddenly we named deletion 22q13, everybody was included. And now there was a system where you had to have SHANK3 to be included. No, now we are including everybody. It helps researchers because they can say, I only want to study the SHANK3 patients, or I want to study the people that don't have SHANK3 involved. Well, now you can say the SHANK3 related or the SHANK3 unrelated. families, same thing for pharma, and same things with families, because families, some of them with interstitial deletions have been told that they don't have Phelan-McDermid syndrome. Well, now, no, yes, you do. You belong to us and we want you. So we've included everyone. And now we're hoping that this will lead to more information about individuals with interstitial deletion who were previously being excluded from the group. We want more information about interstitial deletions so that we can see, is there, as Thomas said, are there genes that have an impact on the interstitial deletions that maybe don't impact the terminal deletions? So we're hoping that this will lead us to more information about Phelan-McDermid syndrome, and we can characterize these groups. So I have given a brief summary of this classification system. I can't make it difficult because it isn't difficult. It's very straightforward and intuitive, and that's all I'm going to say about it. And I will be more confusing on the next topic that I'm going to talk about, which is neurofibromatosis and ring 22. So, and if you have questions about the other, we can talk about that later. So this is very confusing to a lot of people. What does ring 22 have to do with NF2? Well, first, let's talk about NF2. It's a genetic condition that's associated with non-cancerous tumors in the nervous system. So tumors can form on the acoustic nerve that goes between the inner ear and the brain. And if you have tumors here, you can have hearing loss, ringing in the ears, or loss of balance. Well, our kids are often nonverbal or very delayed in speech. So if they can't hear or they have ringing in the ears, how are they going to tell you that? They can't say, you know, they might be banging their head or something like that, but you have to be very vigilant to know that this child has experienced a problem here. And loss of balance, if your child has an unsteady gait or is uncoordinated already, you have to see that suddenly there's been a change in their balance to know that there's a problem, okay? And then tumors can also occur in other places in the brain or any nervous tissue, right? Or the Schwann cells that cover the nerve cells. And sometimes they're on the peripheral nerves, they're on the nerves and on the muscles, any place in the body, and they can occur as subcutaneous lumps on the skin or the trunk of the body. Okay, the next slide. So this slide just summarizes a ring, and I have the diagram that Curtis showed how a ring forms. So it's usually the tips of the chromosomes that are deleted. You're not going to miss half the chromosome and have a ring. You're going to just miss like 22q13, 1213 towards the bottom or towards the top, and it forms a circle, which our brilliant cytogeneticist said, it's a ring, it's a circle, right? It's very intuitive. And then the other two figures show a ring. This is how it looks on the figure on the far left is G banding. It has the banded chromosome, the normal 22, and then the ring. And the figure at the top, that's a different type of banding, C banding, where it just shows the little ring chromosome 22. So that's what a ring looks like. The next slide says, why is there a risk of ring 22? And I have diagrams that will show this, but I'm just going to tell you why. There are two, it's a two-hit activity here, right? The first hit is that the ring is very stable in cell division. So we're talking about mitosis, the type of cell division that happens in your body so that your body can grow. Your cells have to divide. Your child is growing, the cells have to divide so your child can grow and the cells can function, right? So when the cells divide, the ring chromosome is unstable and it doesn't always transfer to the daughter cells as it should. In mitosis, the chromosome chromatids replicate and should produce two identical daughter cells. Rings don't behave like they should. So sometimes one of the daughter cells ends up with only one copy of chromosome 22. The ring doesn't get included and you have monosomy 22. That's the first hit. The second hit is that if in that cell with monosomy 22, the one remaining 22, which is a normal 22, gets a mutation, a pathogenic variant on the NF gene, then you've knocked out both copies of 22 and you're at risk of having neurofibromatosis type 2. So it's two hits. You have to lose the 22 and then you have to have a mutation on the remaining copy of the NF2 gene in a nerve cell. If it happens in a non-nerve cell, then you're not going to have NF2, right? It has to be in the right kind of cell. But let's look at the diagrams and show you what is happening, okay? So this is what happens in cell division. The top diagram A, normal daughter cells. So the little blue circle is a chromatid. It's going to replicate. The little pink circle is a centromere on your chromatid. When the chromatid replicates, you get the blue chromatid and the red chromatid, and they're going to go to two identical daughter cells. And all the other chromosomes are going along with it. I'm just showing you the two rings. They're going to go to two different chromatids, and everybody's happy. But in diagram B, your chromatid replicates, and you can see in the second image, there's a sister chromatid exchange, a little X between the blue and the red circle. And so this is kind of like tying two rubber bands together. When you pull them apart, they break and bad things happen. So in the third image, you have the little blue circle that doesn't have the centromere anymore, doesn't have the little pink thing. So it's an acentric circle, and it's going to be excluded. It's not going to be included in the daughter cell. Then you have the big circle that's the big oval that's blue and red, and it's got two centromeres. So it's a dicentric ring, a double ring. And you see that in the upper pathway, it's going to go to a daughter cell. You have a dicentric ring, a double ring going to a daughter cell. And so that can be bad things. It's basically going to end up with trisomy 22, or it can break and you can end up with two fragments, or you can end up with two abnormal rings, one that's too big and one that's too small. So that's bad stuff up there, right? The other pathway, if the double ring goes up there to one daughter cell, then the other daughter cell on the bottom didn't get a 22, monosomy 22. So that's the first hit. Okay, so this is the abnormal mitosis, right? The second hit is that if you had a cell with monosomy 22, then you have one normal 22 in that cell. And this is your normal 22 in the cell with monosomy. So where is the NF2 gene? This is your normal 22. You can see that SHANK3 is at very bottom of the chromosome, but up about over halfway up the chromosome, that's the NF2 locus. So it's not deleted on the ring. It's up way up there on the chromosome, right? But if you have a mutation of that NF2 locus, that's your second hit. So you have to have the ring. I mean, you have to have monosomy, and then you have to have this mutation. And then you are at risk. It doesn't mean 100% of the time you're going to develop neurofibromatosis. It means you're at risk of developing neurofibromatosis when you have the two hits. So how often does this happen? Let's see the next slide. How many people with ring 22 are going to develop NF2? About 16% of the time. This is a study from individuals at the Clemson University and the Greenwood Genetics Center. It came out in 2020. So you'll recognize some of those names. Curtis is on there, I'm on there, Luigi's on there, other people that you might know. So it's neurofibromatosis in Phelan-McDermid syndrome and ring 22. About 16% of people with ring 22 in this study developed NF2. And the average age of diagnosis was about 18 years. So if you know that your child has a ring 22, you need to start surveillance at about 10 years of age with hearing tests, brain imaging, visual exams, things like that to check for symptoms of NF2. Okay, the next slide. Okay, so in ring 22, the risk of NF2 is not due to deletion of the NF2 locus on chromosome 22, okay? It's not due to inherited mutation of the NF2 gene. So your child didn't inherit the mutation. And it's not because one of the parents has NF2. It's because of what I told you, two hits, error in mitosis, and then a pathogenic mutation. And the reason I'm telling you these three things, and this is in the most cases, because sometimes your child might inherit a deletion of the NF2, or maybe one of the parents will have NF2 and the child has a ring, but that's like not very often. That's like getting struck by lightning, okay? Because when you go to your doctor and say, my child has a ring chromosome and he or she's at risk of having NF2, your doctor might tell you, no, your child's ring doesn't have a deletion of the NF2 locus, or no, we sequenced NF2 and it's normal, so your child's not at risk. Because we have seen doctors tell their families this, and then the family writes to me or Curtis, and we send them literature to take their doctor and explain that, yes, they are at risk because they have a ring chromosome, and you need to watch the child and make sure they don't develop NF2. 16% of the time, not 84% of the time, they're not going to develop NF2, but you want to make sure, right? You want to keep your child safe, okay? Now, the important thing to tell you is that if your child was diagnosed by a microarray, you didn't look at the chromosomes, so you don't know if there's a ring present or not. So the only way you know if there's a ring chromosome is if you did a karyotype and you actually looked at the chromosome. So the picture that Curtis showed you of the karyotype where you saw the chromosomes lined up and the arrow, I don't know if it was a green arrow pointed to the bottom of the 22 and had that big chunk missing, that's how you know whether there was a ring there or not. If you had the diagram where the chromosome was laying down, it had the little purple band at the lower, and it said there was a 4.2 megabase deletion, or it told you what size the deletion is, that's a microarray. The chromosome doesn't give you the size of the deletion. Okay, so if you got a test that told you how many megabases or kilobase your deletion was, that was a microarray. And you might have had a follow-up karyotype, but most likely you didn't, unless you asked for it. So if you didn't have a karyotype, you don't know if your child has a ring or not. You need to have a karyotype to know whether there's a ring or not. Now, if you have a variant of Shank3, not a deletion, then you don't have to worry about this. But if you have a deletion of 22q13 that was diagnosed by microarray, you need to know whether there's a ring present or not. So you're not finished with your genetic studies yet, okay? So that's the bad news. More bad news for today. So I want to impress that upon you, not to scare you, but just to keep your child safe. You need to know that, all right? And then the last thing I'm going to give you is a reference from the NORD, National Organization of Rare Disease. There's a chapter on Ring 22 that Luigi and I wrote, and we wrote it so that we could scare the people with Ring 22 into watching their children for NF2. And so we wrote that chapter. There's also a chapter on Phelan-McDermid syndrome on the NORD website that Kate and Luigi and I wrote. So we wanted to give up-to-date information. We just included in the last 6 months. And that's it. That's all I have.
Thank you so much, Dr. Phelan. I know those are really, really important and sometimes complex topics to describe the shifting classification. I know that's a really important topic for families as well as Ring 22, NF2, and we all really appreciate it. One thing really quick, we are running a little bit behind schedule, so if the panelists wouldn't mind staying after for 10 minutes to get through as many questions as possible, that would be great. If that is not possible, I can try to answer as many rapid fire as I can, and then I'm also writing down all the questions so we can get your answers and send them to you after this session as well. So last but certainly not least, I'd like to introduce Kat Valcourt-Pearce. She is coordinator of publications and managing editor at Gallaudet University's Lauren Clark National Deaf Education Center. I apologize if I mispronounced any of that. Specifically, she works on the Clark Center's Odyssey, which is a magazine that covers new directions in deaf education. Kat and her husband, Larry, are proud deaf parents of 4 young sons, 1 hearing, 3 hard of hearing, one of which you'll be learning about today, Cree, who has Phelan-McDermid syndrome. Kat is also on the board of directors for PMSF and is a strong advocate in our community. Welcome, Kat, and thank you for being here.
Hi, everybody, and welcome. I'm glad to be here. I'm going to share my presentation. Just give me 1 second to pull it up. So I am a parent of a child with Ring 22 and NF2, and I wanted to give you a snapshot today of what that's been like since his diagnosis. First, let me introduce my family. Assuming I can actually, sorry. I'm new at this, so bear with me. So this is my husband, Larry, and our 4 sons, Cole, Cree, Zevin, Kai. The twins are 8, they're fraternal. Cree was diagnosed with Phelan-McDermid syndrome at 4 days old, and my oldest son, Cole, is an OCODA, an only hearing child of deaf adults. We are, I think, the PMSF's only culturally deaf family. And in many cases, NF2 is actually diagnosed through hearing loss, but in Cree's situation, that's not true. That was a hereditary thing. Cree has 22q13 deletion. It's 7.7 to 7.9, and that includes the loss of the Shank3 and the NF2 genes. He was born in DC, and I had a normal pregnancy. He was born at the normal time by C-section, and we knew that he had grade 5 kidney reflux before birth, and he was a little bit floppy, and he was a little bit blue, and he didn't really nurse. He was just tired, and he fell asleep. And a few hours after he was born, they took him to do an x-ray of his kidneys to see how that was going, and they saw a shadow around his heart, and so they called for consult from Children's Hospital cardiology to come. And that night they came to tell me that he had TAPVR, total anomalous pulmonary venous return, and that's a congenital heart defect. So they took him by ambulance to Children's Hospital, and then I didn't see him for about 4 days. So when I was released from my hospital, I went straight to his hospital, and then they first told me he had DiGeorge, and then they said, no, it's not DiGeorge, it's Phelan-McDermid syndrome. And I had no idea what that was. We had no warning of it, and we were completely overwhelmed, and everything hit the fan. And we went home and we started surfing the web, and I found PMSF, and I immediately joined and tried to figure out how we were going to manage the everyday life of Cree and what to expect. My husband, who's a researcher by trade, went to websites and tried to look at all the information on that, research studies and everything. We asked everybody who came in, the 2 geneticists, the doctors, the nurses, the teens, everybody, to try to figure out what was going on. During that time, we had blood tests through Baylor University to check if Cree was de novo or if it was something hereditary, and we found out that it was in fact de novo. It was a fluke, nothing that we passed on. We also found out he was just over a month that he had ring 22 and NF2 through the chromosome analysis and microarray, and that was also a surprise. And so that was the beginning of our learning curve. As I said, we joined PMSF, we started looking around, we read everything we could, and we asked all of those questions. Cree was just over 2 when his first NF2 tumor appeared, and it was on his right shoulder blade. And that was when we started having the annual MRIs for Cree, and we got the NF2 team. We decided to remove the tumor because he was wearing a DMO suit for core strength. DMO suit is like a scuba suit that gives muscle strength, and it kept rubbing against it, and it was causing him irritation. But we didn't know that it was actually causing him pain until after we had it removed, because we would go over a certain speed bump and he was in his car seat and he would cry. And after the surgery, he stopped crying. We're like, oh, and that was really eye-opening for us because we hadn't realized how painful those tumors could be. We unfortunately later had to remove the tumor again, because if you don't get all the cells out when you have the surgery to remove the tumors, they can actually regrow. And Cree also developed other tumors on his back and in other places. This is an example of one of his tumors. That wasn't the one we removed, but it's another one that we had to eventually take care of. Cree's had a couple of other NF2 related surgeries. He had one in 2014. He had tumors on the back of his head, which have actually regrown in recent years. But when he's on the floor on his mat, which is his happy place watching DVDs and stuff, he puts pressure on the back of his head and they grow subcutaneously just under the skin of his scalp. So we took those out. And then we also had one, he had a mass, a tumor cluster in his stomach, and it was growing through his ribs. And so that was also rubbing against the DMO suit. And so we had that removed. We are always in really close contact with the NF2 team, sometimes through email, sometimes at appointments, and we monitor the tumor growth and the impact because they can become really problematic. We don't mess with Cree's tumors unless they're causing issues. In the NF2 community, I call that watch and wait. It's better to avoid surgery as much as you can, because the more surgeries you have, the more chances for complications that can be blood clots and infections and pneumonia, especially for immunocompromised kids like Cree, and then paralysis and even death. But we finally, after watching for a bunch of years, last year decided to go ahead and have this huge tumor cluster removed from Cree's abdomen. And you can see on the picture, it was starting to grow, and you can see the one that was on his side. It just kept growing, and it was getting so big, and it was displacing his liver, and it was making it really hard for him to eat a lot of food. So at age 10, he was only 65 pounds. You can see here, this is a pre-surgery MRI, and you can see where they're measuring the big tumor mass. And then you can also see the white dot in the middle is his spine. So this is him lying down. There's also a couple of other clusters behind those two, that mass cluster, but we left those alone. It was really uncomfortable for Cree. It was very hard for him to move, and it was really heavy. We found out after surgery that it was actually 5 pounds of tumors that they took out. You can see that was in the recovery room. He looks like he was in a sword fight. And then that was the blood draining tube. The other picture is this week, and you can see how much better he looks and how much easier it is for him to move. You can see his open heart surgery scar. You can see the other scar too. They took out 5 pounds of tumors. Now, 1 pound was banked for pathology, for biopsy to make sure it is non-cancerous. NF2 tumors are generally not cancerous, as Katie said, but there is maybe a 5% chance that they could be. So they wanted to rule that out. And then we requested that the 4 pounds of tumors, the other 4 pounds, were banked for NF2 research, because that's how we contribute to the field. Every time that he has any kind of tumor surgery, we ask the tissue be banked for research. Things that you need to be aware of: the tumors can grow anywhere in the body. They tend to be on the brain, the spine, the acoustic nerve, the optic nerves, and then subcutaneous, which is under the skin. The hallmarks of the tumors, they are numerous, they're painful, they're invasive, and our kids have high pain tolerance. We know this, but you can bang one of these things and it will make you cry. It's really painful. You can see some of the examples of the tumors on Cree. He has a very large tumor mass here, and if it were growing inward, it would collapse his lung. We can't remove it because it would wreck his head control, and it would also collapse his right lung, but it's growing outwards, so he's okay. You can also see the one on his jaw. We can't remove that at this point because there's a nerve that runs along the jaw, and if we would take that out, he would probably be paralyzed on that side. So we're not going to do that unless it becomes really problematic, if it's too big, if it starts to hurt. And then you can see the 2 that are on his ankle. And then those at some point we may have to remove because they interfere with his ankle braces. But for now, we're just watching and waiting. Again, the big thing is watch and wait. We wait till we're damned if we're doing, damned if we don't, kind of thing. He has a lot of tumors, maybe 30 now. Some are really fast growing, some are slow growing, some are in clusters, some are single tumors. They're all different. If your child is diagnosed with Ring22, ask for a baseline MRI. You're not going to need the annual MRIs for your child until the first tumor appears, until there are some problematic issues, or if your child is having deafness issues and you don't have any explanation for it. But you want to get a baseline MRI once it's diagnosed, just so you have something to compare it to. As Katie said, PMS is not associated with Phelan-McDermid syndrome, only Ring22. You can have NF2 on its own, and it's often inherited in most cases. That's not the case with Ring22-related NF2. And the ring is unstable, so while your child is not always going to develop NF2, it is a possibility. You do want to keep an eye on it. Just keep it in the back of your mind. And then, let's see, lifespan, clinical trials. It used to be that the lifespan of a person diagnosed with NF2 alone was about 15 years from diagnosis. And most people were diagnosed in late teens, early 20s. And that was generally because all of a sudden they had a hearing loss. But with Cree, we don't know. He was diagnosed at birth almost with both. He was a newborn. And so for him, you really can't say what his lifespan will be. You just hope that he lives as long and as happy as he can. People are living longer now. It depends on, of course, the additional medical conditions, the number of surgeries they've had, and other factors. Are they immunocompromised? Anything like that. When Cree was diagnosed with PMS, there were about 800 people worldwide with that particular condition. Now there are probably fewer than 2,000. I'm not sure the exact number. Currently, worldwide, there are just over maybe 100, I think, individuals diagnosed with Ring22, and even fewer diagnosed with Ring22 and NF2. And there's really not much information or research studies that exist on these conditions together. I know of a handful of individuals that have both. There are very few clinical trials out there for NF2 patients of any age, and there's no cure at this time. These are some of the websites that help me if you want to get some more information. Obviously, PMSF, the Neurofibromatosis Network, Children's Tumor Foundation, RARE, and also NF Hope does fundraising for NF research. My email for questions, if you have any, you can always contact me. I'm on Facebook. You can always direct message me, private message me. And I really thank you for your interest. And if you have any questions, please put them in the chat on there.
Thank you so much, Kat. Thank you for sharing your beautiful family with us and your story. And I'm sure it brings comfort to others to see everything that you have done, and also shared resources with everybody. So we really appreciate that. As I mentioned, we are a little bit over time. It is 2 o'clock on the dot right now. The panelists have been very gracious and willing to stay until about 2:15, and we will cut it off there. But if anyone is still hanging around and wants to listen to a few questions answered, please feel free. We will also make sure all your questions get answered after the fact, and possibly even put out a Q&A with the answered questions later. First off, we received a few questions about Thomas' talk, specifically about lithium. I would encourage anyone with any questions about clinical trials to go to clinicaltrials.gov and search for Phelan-McDermid syndrome, where you can find all of the information about what is enrolling and when, and inclusion criteria. We received one question about why is the age limit 18 years of age for being included in clinical trials. This is not always the case. Each trial is different. So age of enrollment, whether on the lower end or the upper end, changes every time. And this has to do with a lot of factors, but the study personnel often have reasons for this associated with when they think something will be most effective. Okay, so I have narrowed this down to just one question per panelist, and then there's one more, philosophical one at the end that I would like to take a stab at. First, for Curtis, we got a couple of questions in the chat that follow the same theme of asking about their own child having a specific variant in SHANK3. And I think it brings up the question of, what does this term pathogenic variant mean? And how does that differentiate from some other change to SHANK3? Are all changes to SHANK3 causative of PMS?
The short answer to that question is no. We find variants in genes all the time, and all of us carry variants in a variety of genes in our body. The determination of pathogenic is made first in the laboratory by the molecular scientists who are in the laboratory, and they have a specific set of guidelines that they use based on the American College of Medical Genetics. And using those guidelines, they basically have a scoring system, and you have to check so many boxes for that to be pathogenic. We have 5 categories: pathogenic, likely pathogenic, variant of uncertain significance, likely benign, and benign. And based on the scoring system, they classify things into those categories. I think the clinical part comes in is based on their scoring system. Sometimes they cannot score something as pathogenic or likely pathogenic. However, we're looking at the patient clinically, and the features that we're seeing clinically in the patient certainly support that being pathogenic. Remember that these are all, I won't say all, but many of these are individual changes present only in that particular person. If we have another individual who has the same change in that gene and fits Phelan, we'll use SHANK3, fits Phelan-McDermid syndrome, then that lends strength in the molecular lab to say this is pathogenic. But sometimes we as clinicians will say, well, I know what the lab is saying. They're saying this is a variant of uncertain significance. However, I'm looking at this child or adult, and I feel this child fits this, so I think this is likely pathogenic. Then we get into a quandary of trying to prove that that is pathogenic, and that is going to take functional studies. In other words, testing cells from that child or putting that variant into an animal model and demonstrating conclusively that this leads to abnormal synaptic function, abnormal behavior, or whatever else that lab may be measuring. So that can be tough. And it's a, you could take one variant in one gene and you could make a PhD thesis out of studying that. So it can be extremely time consuming.
Absolutely. Thank you for that answer. And that also points to a different question in the chat, which can be summarized basically as asking, and how come people with the same genetic alterations or very similar genetic alterations could have very different symptoms, which could occur for a number of reasons, environmental factors, age, many other things going on. So we know that that is also at play.
Right,
Katy.
many other genetic factors. Again, if we're talking about a deletion of 22 or a pathogenic variant of SHANK3, we cannot ignore those other 45 chromosomes that are present there besides that 22 that has the problem, and all the genes that are contained on there, and how they could be impacting what we see as the clinical phenotype.
Absolutely. And I know you've been instrumental in helping some families understand maybe some of the other genetic changes they have that are outside of the 22q13 region. One question for Dr. Phelan. Do you need to do additional genetic testing for NF2 if the deletion is a result of a translocation?
No, if it's a translocation and it's not a ring chromosome, then I wouldn't worry about it. No, it's only the instability of the ring chromosome that lends itself to the risk for NF2.
Thank you. And a question for Kat. Thank you for sharing your story. How do you estimate pain associated with Cree's tumors? Are you able to gauge this?
You know, it's really hard because he's nonverbal and he can't point to it. And so sometimes I just press on them. I make a habit of pressing on tumors and seeing if he flinches, because that's the only way I know his finger one can be painful. I know his tumor mass bothered him because he would have such a hard time moving. So you just kind of have to watch and try to figure out signals. There's no good way to do it. And he may have other pain that I don't know about. And it's really hard as a parent because you want to solve everything. And sometimes you just can't. You have to figure it out as you go.
Let me say, as a clinician, I find it not wise to, let's call it unwise, to argue with a parent who is living with that child about their child's symptoms, because they are there every day and I am not. And if you think you don't want to cross a parent, you sure do not want to cross a grandparent, especially a grandmother.
Wise advice. Okay, so the last question that I wanted to get to, and I do promise that we will address all questions and we will send them out later. I thought this one was a particularly beautiful question that branches into advocacy. It says, it's obvious that more genetic testing needs to be done on individuals with delays and those with diagnoses of autism. I also know how hard it is to get the right testing done if you don't have obvious or severe characteristics or symptoms. How is that being addressed? How can testing become more accessible when there's so many hurdles with insurance and the financial impacts these tests have? I just had a few comments on this, and of course, Curtis or anybody, please jump in if you have additional comments, but I think this will be addressed in a couple of ways over a long period of time. One is the advance of technology. So as technology advances, which is a more passive way this will be addressed, sequencing and other forms of genetic testing will become less expensive, and therefore it will become more of a standard to do certain tests first that were otherwise possibly prohibitively expensive. I think the other piece to that is advocacy, and also really having good ways of collecting data in the syndrome and in autism. So advocacy, there are a number of groups that we work alongside, and we will have an advocacy arm to this conference where we will give everyone opportunities to sign up or attend sessions about what we can all do to change policy, including just putting some effort into how we communicate with legislators. There's also things like applying for an ICD code, which can help gather information that's really important for insurance coverage and other things which could lessen the burden. And the final thing is socioeconomic diversity being a really big challenge to getting great healthcare all the time. And that is another area where I think a lot of groups and advocacy is generally moving in to make these things easier. And I think it will unfortunately take time, but I think it's a theme that a lot of people are really invested in right now.
I would just add to that, this is the importance of participating in the registry and in the foundation, because it gives us, the more folks who are involved, it gives us more oomph and push for legislators locally and nationally and internationally and other places to advocate for testing. And remember, most of the times when we go in testing a child with autism or developmental delay, we are not testing specifically for Phelan-McDermid syndrome. We may find that, but we're trying to find the etiology for that family, whatever it may be, so that we can start addressing that child's needs and hopefully offer them the opportunity to be enrolled in a clinical trial if that is available. So it's not just finding PMS, it's finding the genetic cause of autism and intellectual disability. And what we're going to find, I think, is that it is a combination of many genetic factors, multiple variants and multiple genes, plus environmental factors that may play a role and have an additive or synergistic effect in getting that diagnosis. But that makes it even tougher to do when you don't have a specific gene with a change in it or deletion of 22q13.3. When we start trying to add all of these things together, it becomes incredibly complex and difficult.
Thank you. That's a very important point. So we are at right at 2:15. We apologize for going over. We wanted to give such a sincere thank you to our panelists. They have been just absolutely amazing working through this with us as the first session. They've each created brand new presentations for this, and they all have expertise that we hope was displayed here today and investment in all of you. So thank you all, and to the conference planning team, and we will see you for the next session. Thank you.
Thanks to you, Kate. We appreciate you.
Bye-bye.
Don't forget to tune into our next session, The Good, the Bad, and the Funny, a stress relief session for caregivers presented by Katy Phelan on Saturday, April 9th at 7 p.m. Keep your eye on your app to learn more details. This session won't be recorded. So if you want to participate, we look forward to you being there and sharing your stories. Thank you.
All right. Bye-bye.
Bye.
Bye.